take you guys away from the command line for a short period of time, give you a break from typing in code, and I'm going to talk about what you do once you have your genome. So you have an annotated genome, and of course you could do a head of your file and be very proud that you have a nice FASTA sequence with a chromosome ID to it. But that isn't really, I mean, obviously you're generating a genome and you're annotating that genome because you want to do some analyses on it. Such as, you want to compare genome organization between species, you want to visualize the genome and its functional elements, et cetera. You want to blast it. You just want to see, like, is my favorite gene or a set of genes present? Or, you're sequencing it for the sake of sequencing it. And you just want to have a public identifier that you can, put in the paper when it comes time for publication. So the resource, the tool or set of tools that I'm going to talk about today are Koji. So Koji is this comparative genomics website at genomeevolution.org. Don't make the mistakes of having don't make the mistake of having two E's. It's confusing. But Koji, for one, it's a great resource for depositing and searching through genomes. There are roughly 54,000 genomes currently present, or stored in Koji. Many of those are different variants of the same species, but there are roughly 20,000 organisms for which we have genomes present in Koji. And you can do a lot of things with those genomes. And I'm going to talk about some of those things today. So we'll talk about each of these different tools or aspects of Koji, starting with how to get your genome from Cybers into Koji. Now, there's a tutorial on our GitHub that kind of talks point by point how to move files into the correct location or do different things with CyberDuck. But I'm just going to, sorry, I need to get a new tab. Okay, so first we're going to go to the DE. And we are going to go to where our data are stored. And here you may see that you have a Koji data folder. If you do not have a Koji data folder, then you need to go to this link, user.cybers.org/services/mine. Oh, come on. And right down here where it says My Services, you should see there's an option, or actually it's probably under Available. There's an option for Koji, and you'll just have to press Request Access if you don't already have access. I already have access with this user account, but you might. So if you don't see that you have a Koji data folder, then you'll need to activate the connection between Koji and Cybers. So Koji and Cybers are essentially, the data are stored in the same place, and there's a nice symbolic link between the two databases or the two resources. Okay, so once you've activated this, you'll refresh your discovery environments window, and you should then see a Koji data folder. And what you can do is you can go into folders where you have stored your genomes of interest. You can click on those genomes and maybe the associated genome annotation file. This is for Utricularia. And you can go to these little three dots and you can just say, let's move. And where do you want to move them to? You want to move them to the Koji data folder. Now, I've already done this. I already have Utricularia present in this folder. So, well, do I? Oh, it's in this workshop folder. So I'm not going to do that. But you would move your genome and your genome annotation or whatever genome files you wanted to have access to from Koji. You would move that into the Koji data folder. Okay, so I'm going to cancel that. Now, everything that I have in this Koji data folder, just take a mental snapshot of what's here, because we're going to go back to the Koji website, and we're going to go to, so once you've signed in, you'll sign in over here on the right. You'll go to My Data, and once you're in My Data, over here on the left, you have these options for adding new things or creating new things. Now, there are different things that you can create. You can create organizational items like notebooks and user groups. You can also add in experiments. So you can add in RNA-seq data in here to visualize within a genome browser. But here, we're going to add in a new genome, so that you can start playing around with whatever genome you may have just created. Now, this process is pretty straightforward. You just have to fill in all of the asterisk items. So for here, we're going to be adding in Utricularia gibba. And there's already a Utricularia gibba in the database. So this is allowing us to skip a couple of, shall we say, metadata steps that Koji usually asks for. But if it already knows what species you're asking for, then you don't have to do any tax or add any additional taxonomic information. But if I were to say, let's add Andrew Nelsonicus domesticus, then it's gonna be like, okay, there's nothing here. You're gonna have to add in a new species, and so you would have to go in and grab the taxonomic information from NCBI, and so you would just find the closest species of interest to the one you just sequenced, and you would bring that in. I'd say I'm a Neanderthal. There we go, and then you just create. But again, Utricularia is already in here. So an important thing about the taxonomic metadata is that it's important if people are trying to find a genome, or so maybe they're trying to find something closely related to what you're uploading. They don't know that you've actually uploaded and made public this particular genome. But if they search under, say, angiosperms, and they search under rosids, and they search under brassicaceae, everything that falls under that lineage would be returned in their search. And so they could identify what species, they could identify your genome by searching, because the taxonomic information is there. Okay, Utricularia. This version should correspond, I mean, since you are the one making or assembling and annotating this genome, you get to do the versioning yourself. Typically, this would probably, if it's the first assembly and it's the first pass through, it's probably going to be a number less than 1. But honestly, you can have this version be whatever you want. But the next time you upload a different version of this genome, you would want to have it be somewhat later in the sequence. So if you do 1 for the first time, you do 2 or 1.2 for the second time, whatever. Okay. So this is essentially unmasked sequencing data. I think there might be masked. Yeah, so you can add in masked sequencing data, hard masked by repeat master. I think that's what Susie used. We can toss that in. Source, this can be whatever source you want. You're kind of either giving credits to another organization if this is a publicly available genome, say you got off at NCBI or you got from a publication, or you can credit yourself because you're the one that sequenced and assembled, et cetera. So I'm just going to call BTI workshop. Oops. Okay, So you might have noticed that this restricted option is automatically enabled. And that means that the genome that you upload is restricted to you and whomever you choose to give access to, unless you unclick this option. So this means that you now can look at this genome. You can do all sorts of analyses with this genome and share this genome with other people, but nobody else can, except for the admins of the system, me and air clients. But nobody else can look at it unless you give them access. So that's cool. That means you can kind of play around with your genome. You can kind of do some sanity testing, all these other things. You can analyze it ad nauseum until you're ready to publish and make it public. You can add in additional descriptions, other types of information. None of that's super necessary, but if you have it, you might add it. Okay, sorry, I'm just now seeing messages. Having trouble getting the Koji folder in the DE. Can someone say again what I need to do to get access? So you should go, I'm going to copy this into the chat. Go here. And if you go there and you log in, you should see Koji underneath the available services. There's a whole bunch of services here, but the one you want is Koji. And then you'll just click request access. It's an automatic thing. You may have to log in again. I can't remember. And then you'll be able to go immediately from there. Okay, so back to I'm trying to select data to pull in the genome of interest. And you may notice that the folder in the discovery environment looks exactly the same. That's because this is the Koji data folder. Importantly, this is the only folder that Koji has access to within your Cypress data store. So this is kind of a security feature. So you have to move things into the Koji data folder so that Koji can actually see those data. Okay, so I'm going to pull in the utricularia genome. This is in the workshop folder that I already created. And I'm looking. Let's do UGIBA. Sure, let's do that. That's kind of small, so that's probably just a subset of the utricularia genome. Okay, so I told it where to grab the file from. You can also pull in, you can upload from your desktop. You can also pull in from an FTP or HTTP. So any kind of publicly available genome, NCBI, phytosome, etc. You can easily pull in. But anything that's on phytosome or NCBI is probably already in Koji. So if you don't, I would search for it. If it's a publicly available genome, I would search for it first. Okay, we'll start that. This should come in pretty fast. I mean, it's just a single, actually it's just a few chromosomes. Okay, and then we can go to the genome info page. Okay, so here is the kind of the base information for the small portion of the utricularia GIBA genome that we pulled in. We can get a sense of how long these chromosomes are. You see, we can get an N50, et cetera. This obviously is just a subset. So our N50 is pretty awesome. I would be happy with this, especially from a first pass. But it also gives us other information, features, et cetera. So the very next thing we would need to do is we would need to associate a genome annotation with this genome. Now, you would have gone through, how do we get the data into the Koji data folder? Okay. So back in your discovery environment, if you go to your data folder, this is kind of your root directory, you're going to have a number of folders, mainly, well, if you've done anything in the discovery environment, you will have an analyses folder, you'll have some kind of data folder. Here it's just genomes I love. I don't love very many genomes, clearly. And then you'll have the Koji data folder. So anywhere you have a file, you can just click on that. You can actually click on multiple, and once you've clicked on both of those, you can go up here and move them using this this move button. So go up to the 3 little dots and move. You can move to a specific location and just go to the Koji data. If you do not have any, if you don't have any genomes, you can go to the, and I can't remember if these data have been shared through the discovery environment or through the data store, but you can go to shared with me, and I have a whole lot of things shared with me. Oh, no, that's not shared with me, it's community data, sorry. And then I think, Susie, do you remember where the workshop stuff is? It might be in training.
I thought
it's right here.
Yep.
Sorry, I just passed right over. So Botany 2020 NMG workshop. You can go in here to, we'll say assemblies, UGIBA. I think this is actually, yeah, this is the full genome, that like the published genome. So you can oh whoops, I'm actually sorry, I'm actually trying to move things to that location. Anyway, you can copy from this Botany 2020 workshop into your Koji data folder, and you can play around with that file instead. Okay, come on, go away, Zoom. I hate how the Zoom like controls will pop down right where you're trying to, oops, that's not the one. Okay, so I'm actually going to look at a different utricularia that I loaded up a long time ago, because it already has the genome annotation. But I'll show you how to pull in the annotation, but we won't go through that because that takes a few minutes. So this is the same view we were just at. We were looking at the genome. We were looking at all the genome information. The very first thing we would do after we pull in a genome is we would assign, or we would add in genome annotation for it. So pops in a little help info. This is the genome. This is the name for the genome that we're looking at. We can again assign a version. We can assign a source. TI something workshop. That's fine. Then going back to the data store, I'm in my Koji data folder, and I would just go to utricularia, and then this GFF is associated with this genome. Okay, so I have actually already done this, so I'm not going to do that. I'm going to go back to this genome, and we're now going to look at the features, and now we see that we have not only do we have chromosomes, but we also have roughly 30,000 protein coding genes, which seems about reasonable. Kind of the standard diploid state of genes is about 24K or so. So this looks pretty good. I don't know anything about utricularia's ploidy level or any of that stuff, but that certainly seems like a good set of protein coding genes. So where do we go from here? Well, the next thing that we can do is we can browse this genome. And if you click on the browse button, it pops up your standard kind of genome browser view. And I saved something I had in from before. So this is kind of something you'll be familiar with if you've ever looked at a genome browser. You just have the coordinates along the chromosome that we're looking at, and you have all the annotated genes. So one thing that you can do with Koji is you can pull in, RNA-seq experiments. You can pull in, epigenetic experiments. You can pull in essentially anything that is mapped to your genome, and then you can overlay that. And we actually have a lot of utricularia. That one's not it. Have to find the right. I shouldn't have killed that. Okay, there's no. Anyway, there we go. There's the so now we have RNA sequencing data, the same RNA sequencing data that we use to annotate the genes or help to annotate the genes for this genome. We can now visualize where that is, where those RNA sequencing data or those reads are mapped to. And so standard type of stuff, you can zoom in, you can see, okay, this gene, well, this gene's pretty highly expressed. We can kind of have normalized counts of how highly expressed this gene is relative to others, et cetera. We can also click on these features. So I clicked, whoops. We can click on these features and we can look at particular information associated with them. We can go and grab the FASTA sequence. We can port them directly into Koji Blast, and we can see what sequence similarity or what other genes these things are present in utricularia or other genomes that are similar to the sequence. So let's do that next. Let's take this kind of highly expressed gene. We don't know what it is, and let's take it and send it to Koji Blast, and let's see if it is similar to anything that maybe we have an annotation for. So I'm going to start out with Arabidopsis thaliana, even though it's like extremely unrelated to utricularia, simply because it has a really good annotation set. Is there any other genome that would be closely related to utricularia that might be better annotated? What's the family that utricularia is in?
Oh, I don't remember off the top of my head.
Let's go back. I can answer that. It is in, where is this?
Well, then I don't even know how you say this here.
Fibulariaceae. There's another carnivorous plant I thought that was sequenced.
Okay, so Lamiales. Let's look. Okay, let's see here. Where's my Koji Blast? Okay, let's add in Lamiales and see what pops up. Okay, so now this is getting it and why it's nice to have that taxonomic information, because now we can go through and we can pick other things that fall within the family, and we can just blast our whatever gene we've just identified that's highly expressed in leaf tissue, and we can blast it to those other species. So there's a lot of stuff on here that's locked, so you actually wouldn't be able to see it because it's all private. Probably releasing trade secrets by okay, let's pick this one. Okay, we'll set the E value kind of low, and we'll let the blast go and see if we find anything. I do not know what Interinum majus is.
Snapdragon.
Oh, there we go. Okay, so whatever gene I picked, luckily and this was completely random, I promise. Luckily, whatever gene I picked actually has some kind of sequence homolog and a pretty good sequence homolog in Arabidopsis. That actually has multiple. So let's click on this and let's see. Okay, so the sequence I selected is actually similar to the third and fourth exons of this particular gene. Now, if we go back, where I think I probably closed this, but you'll remember that this gene was only a 2 exon gene in Utricularia. So this could be just a shared domain, or this could be that the Utricularia gene itself wasn't a complete annotation. There are a number of ways in which we could look at that and determine that, but they might be out of the scope of the time that I have today. But let's click on this. We can click on this Arabidopsis gene either here in this view, or we can click on it here, and we can just see what is this thing. So this is some kind of glycosyl transferase galactanol synthase. Don't know exactly, but it sounds very metabolic-y, which is what I was expecting, because it's leaf tissue and it's highly expressed. So the main things that are going to be highly expressed in leaf tissue are probably going to be photosynthetic related or metabolism related. So that's what I was going for. I was hoping to find something that was conserved across long distances. Okay, we can also look at this this hit in Snapdragon, the top hit. We see, okay, this is just a single exon hit. Yeah, same here. So probably what we're identifying from Utricularia is just a conserved domain from in that gene from Utricularia with these other 2 species. So it may not be a glycosyl transferase or whatever, but it's probably something to do with I'm guessing metabolism. Okay, so let's do something additional here. Let's add back in Utricularia. This one. We'll wait for it to load because there are a bunch. We've got to go through a whole bunch of auto-generated something or other. And then I was hoping to find the one that I created. Well, that one will work. Okay. And then we're going to run this again. And then we're going to look at conservation of the regions around the genes. So we know that this gene is at least parts of this gene are conserved. But let's see if there's anything deeper. There's any deeper conservation between these genes. Well, we can be pretty certain that there's not with the Arabidopsis, but maybe there is between Utricularia and Snapdragon. So let's take this to GEVO. And what GEVO is going to do is GEVO is a very kind of, I'd say, a fine scale synteny. It's looking at synteny between the genomic regions between these 3 genomes at this particular location. Now, it's not looking at whole chromosome synteny. It's just looking at conservation of a few genes around on either side of our target gene. 50,000. Actually, let's do 100. Okay, so I'm just making the window in which we look a little bit bigger because I'm not actually expecting much conservation between even Snapdragon and Utricularia. So essentially what this is doing is just doing a large scale blast or a last between each of the other regions. And you see here in yellow is our kind of focal gene or the hit of interest. And you see, so Arabidopsis, Snapdragon, and Utricularia. And too zoomed out. The graphics on this. Anyway, you see that there's very little sequence very little synteny. In fact, there's no synteny, I would say, between Utricularia, this one on the bottom, and the Arabidopsis genome and the Snapdragon genome. And again, this is partly to be expected. We weren't even sure if these sequences were orthologs. They just share some kind of sequence similarity. But this is just an example of a couple of things you can do very quickly after loading in your genome of interest. Now, let's go back to my slides. So thought we'd talk about loading genomes, organism view, Epic Koji genome browser. Then we kind of briefly talked about Koji Blast. Now let's look at a kind of large scale synteny. And to do that, I'm going to go, sorry, let's close this, tools. We're gonna go to SynMap now. And just to like have a better demonstration, I'm gonna pick two very, very simple genomes for which we know a lot about what has occurred, or the relationship between these two genomes and what has occurred between them since they last shared a common ancestor. So let's look at Arabidopsis thaliana. And we're going to go to Colombia. And then we're going to do Brassica rapa. Okay. And then I'm going to clean this up a little bit just to make it look nicer. Okay. Now I think I already have this run in one of these other tabs, but I'm just going to let this sit for a second. So what this is doing is, again, this is doing essentially a genome against genome, or a whole genome last or blast to see like how things line up, and then it's checking to see if the different regions retain synteny, and if there are any kind of like large duplications of large chromosomal blocks. So that ran very quickly because I've already run it. It's essentially stored in memory. There's some information there about what's going on. Oof. Okay, so the very first thing that we can see is this dot plot, essentially, that if you have picked two random genomes, you're going to have one genome on the x-axis, another genome on the y-axis. And what you should be able to see here is that for any region within the Arabidopsis, for any region on the Arabidopsis chromosome, there's one or multiple corresponding regions for Brassica rapa. So if I were to like move the line across, we see that chromosome 1, there are several sections where there's been either some rearrangements or there's been some minor duplications. And like here's a nice instance of a rearrangement that occurred in the last between these two genomes. I need to check one thing. So change that. Let's run this one more time. So essentially, what was occurring is the SynMap was only recovering, or was only displaying the primary ortholog, and it wasn't showing the other synthologs that I was expecting. So this is just going to run one more time. I might have it. Yeah, I have it here. But anyway, okay. And while this is running, actually, never mind. It's already done. Okay, got it. Okay, now it looks a little bit messier, but now it actually has everything that I would expect given the relationship between Arabidopsis and Brassica rapa. So now I can explain this in a little bit more detail for those of you who are not familiar with the most important plant family in the world. Kidding, Susie. So Brassica rapa is what we call, I think it's a mesopolyploid. It's undergone a hexaploidy. So it has since it last shared a common ancestor with Arabidopsis. Arabidopsis itself has undergone a rearrangement, that is a genome rearrangement process that makes it look kind of weird relative to the kind of the standard karyotype for the family. But the important thing is that Brassica rapa should have 3 copies of most genome segments relative to Arabidopsis. And so now if I kind of hover over and I move across, you see here on chromosome 1, you see that there's actually 3 segments that map back to chromosome 1 in Brassica. Right here, and then a kind of some kind of inversion and some weird events that happened here. And that should be mostly the case all the way across. Now, you may notice, or I mean, if you're looking through this, and you know, some people are better at looking at these workshop lots than others, but you may notice that there are certain sections that don't have 3 copies. And that is because of a phenomenon that some of you may have heard of called fractionation. Essentially where, like, post-polyploidy, bits and pieces of bits and pieces of the genome are going to be lost because they're sort of not really important, they're redundant, maybe they belong to what we would call a weaker subgenome than the others. And I'm butchering a lot of evolutionary theory here. But suffice it to say, if you're interested in this fractionation process, I turned on this, this feature that allows you to see which of the Brassica subgenomes, or which of the Brassica chromosomes line up to each of the Arabidopsis chromosomes. And it's a little bit messy here. It's not like some comparisons are much simpler because, say you're looking at the diploid progenitor species for Brassica, then it would be a lot cleaner because you would see 3 lines for each Brassica. Yeah, you would see 3 lines for each comparison. But here, you not only have evolutionary events that happen to Brassica, including this polyploidy, but you also have evolutionary and genomic events that happen to Arabidopsis. But if you scroll across, this is, again, chromosome 1 in Arabidopsis. You see that here there's really 2 regions that share a lot of similarity to the Arabidopsis chromosome 1. And then you see like these interesting inversions where, so we were comparing chromosome 7 and 2 of Brassica, but then you have this weird little bloop where now chromosome 9 from Brassica has that information, that chromosome, that information that's homologous to the chromosome 1. And again, you'd have to look into this further, but this is probably a genome rearrangement event that happens specific to Arabidopsis. Because Brassica, for the most part, has retained kind of a similar chromosomal structure to most other Brassicaceae. Arabidopsis is the weird one out. Here on the right arm of chromosome 2, you see this nice, you see these 3 subgenomes, these 3 chromosomes within Brassica kind of competing for conservation. So what this is actually showing is kind of the percent of genes that are retained within a specific window. And so you see like, okay, this blue chromosome, chromosome 5, retains most of the synthalogs, most of the genes on the right arm of chromosome 2. but you see that that isn't always the case. There's these subgenomes are kind of battling it out with each other for bits and pieces of the Arabidopsis chromosome 2, whether or not they are retained or not. Anyway, I think this is a particularly fascinating thing to look at, especially, I mean, it's really important following polyploidy events. But you can look at any relatives. If we were to look at, I think I have, yeah, this is Arabidopsis lyrata and compare it to Thaliana. So lyrata has not had a duplication or a polyploidy event of any means. And lyrata represents kind of the more typical karyotype for the family. And so you can actually see the rearrangement events that happens in Arabidopsis thaliana. So Thaliana had rearrangement and condensation. And so what used to be 8 chromosomes, and is 8 chromosomes for most species within the family, for Arabidopsis, our most esteemed model organism, which happens to have a very strange genome, there's only 5 chromosomes. And so you see a lot of the kind of evidence of that interesting, those rearrangements. You see a rearrangement here, or an inversion, sorry. You see like some of these chromosomes are actually been split apart or merged together, depending on your perspective. And again, you can kind of see that in these fractionation bias plots. So here, Arabidopsis lyrata chromosome 3, much of it matches to Thaliana chromosome 3, but then towards the right arm, you see this flips. Okay, so let's see, did I cover all the important bits? Yeah, I feel like I kind of went through that quickly, but you can blast, you can look at genome evolution, you can compare between species, you can just make your genome public, you can make it viewable by other people. And if you have any questions, you just email down at the bottom of the Koji. You can just click on the contact us. That emails straight to genome.koji.gmail. And Eric or I will answer. If it's an easy question, I'll answer it. If it's a database question, then Eric will answer it. Okay, I hope that I've given you plenty of time, Adrian, to talk SNPs, talk something more substantial and important.
I don't know about more substantial, more important, but something different.
I had a quick question, Andrew. Are those fractionation plots new? I don't remember those being there before. Maybe I just didn't look around enough.
They've been here for a while, but they only sorry, let me share my screen one more time. They only get turned on if you go into the analysis options and you click run fractionation bias.
Ah, okay.
Yeah, otherwise it's automatically turned off. You can also turn on, you can also look at the K/N/Ks ratios between syntenic coding regions by turning on these values here.
That I knew about, I think.
Okay,
but yeah,
okay,
I did not know about those plots. So that is good to know. Thank you.
Yep. It's something fun. It's yeah.
Are there any questions for Andrew before we turn it over to Adrian?
Okay.
Thank you, Andrew.
Yeah, thanks.
No problem.
So I will share my screen. I'll share this desktop here, I think, again. So I guess the other kind of component that we wanted to touch on a bit is kind of extensions on SNP calling. So a bit about SNP calling and kind of extensions of SNP calling. And actually, I will put this over here for a second. So this is the GitHub page. And so if you're in this repo and you click on 6 downstream analyses, there's a folder for Andrew's materials here. And there are 2 additional PDF files. One is the slides that I'll be going through and talking through for this section. And then there's this other PDF called SNP Calling 2021 PDF. And so basically what I'm going to go through is primarily a module that was developed by Jacob. And this document here contains all the kind of detailed instructions for kind of going through this module. We won't go through all the commands today. So what I mean, most of these commands take a fairly long time to run with the data sets that we have. So we'll kind of leave that for you for homework exercise that you can sort of work through on your own. What I do want to go through in terms of the slides is at least talk through the kind of main sections or components of what you'll be going through. And initially I had, when people started signing up, they were mostly people who had just taken our BTI bioinformatics intro course. So the way I sort of set up the slides was to kind of show how they were extensions of the SNP calling that we did in that course and kind of built on some of the elements in there. But I think the main goal is to provide context for everyone, whether you were part of the course, whether you were fortunate enough to be part of our course or not.
I just wanted to point out real quick too, the materials for the course are available online. So if anyone wants those, they're actually on our BTI BCBC website, or you can email any of us and we can point you to it.
Yes, definitely. For those who weren't in the course, we have the materials up there as well. Yeah, maybe I can, before we end, I'll grab the link and maybe put it in the chat as well. So the SNP calling components that I wanted to talk about, it includes SNP calling, but there are a number of kind of branch points from there as well. So the main idea here is, now that we have a genome for our non-model organism, we can generate sequence for a number of other individuals in the species. And there are lots of different options: RAD-seq, HIB-seq, RNA-seq, whole genome sequencing, GBS type approaches. And so this sort of enables you to, once you have the reference genome for your non-model species, to go out and in a relatively a more inexpensive way, kind of do a lot of sequencing for individuals in that species, and then do these sort of population genetic analyses. I will make some reference as well, and it's covered in sort of the detailed instructions. There are options for de novo clustering of sequence data, even if you don't have the reference. I think most of the examples focus on having a reference because with Jacob and Susie's assistance, you have now developed your reference for your non-model species. So you should be okay on that front.
Or they're so terrified that they'll never try this again.
They're all just going to work on Arabidopsis for the rest of their lives. No, that would be so sad. No, we've got our reference.
I take offense. I thought Arabidopsis was the best model organism ever.
Yeah. Well, you can dish it out. I can dish it out too.
It's a crappy model.
Yeah. I mean, it served its purpose. Blah, blah, insert diplomatic statements here. Yeah. But so in keeping with the theme, the non-model theme of the workshop, so we can generate this reference and do some SNP calling, kind of leverage this for studying the diversity within our species. And so there are kind of, I guess, four main components that are covered in this module. So the first is mapping our reads. In this case, they're transcriptome reads to the reference genome. Calling SNPs, the sort of key step in terms of determining what variants we have in our individuals. There's some filtering steps as well, primarily using a tool called VCF tools, or a program called VCF tools. And then a step where you can generate a PCA, which is a common means of so doing the principal component analysis, that's what PCA stands for, and then often generating a plot with usually the first two principal components. And so in that case, each dot would represent one of your samples, one of your individuals, and they would cluster according to their degree of genetic similarity or difference. And so I'll just kind of walk through some of the commands. Obviously, I'll expect, well, if you were so inclined, you would go through these on your own later, but I want to give you a sense of kind of the main steps, the main things that are happening. So one of the things, you probably have done this in Jacob's session, is installing BWA. But if you didn't do that at that time, you'd want to do this in your atmosphere instance. And so in terms of the first step, so this involves mapping reads to the reference. And you see, I included some of the CD and CP commands in here. Again, the full details of the various steps and your locations are in the other PDF document. So the first step is we're going to use BWA to index our reference genome. And again, indexing of a reference is essentially a step that will speed up subsequent processes, particularly the mapping. So I usually mention the idea that the index, when you're trying to search for the location of something, just as you would in a book, you don't necessarily look for all instances of a given word by sort of starting on page 1 and working your way through. If you're looking for a specific word, you go to the table of, or the index at the back of the book and sort of look through I, N, D, if I were looking for the word index. And that sort of speeds up the process of finding where a given word is. And so the same sort of concept applies in the case of a genome index as well. And so once we've done the indexing, we'll proceed to this script called bwa_ugiba.sh. And again, this script is in your Scratch your folder that we attached to Scratch or copied over to Scratch. Scripts, again, are essentially just text files with lines of code, right? So you can look in that script by using either Emacs or less. I think we used Emacs earlier to open things up. Less is another command that you can use to look inside things. And so it's just a text file. It has a number of different commands. But just to give you a sense, so to run it, you would just use this command here. But to give you a sense of what's inside that, I would encourage you to look in The main kind of command that's doing the mapping is this bwa mem command. We pass it a number of options. This is the number of threads to use. This is sort of adjusting the information of what are called the read groups. So it's just sort of specifying or adjusting the types of information that are included in these read groups for the different reads. We tell it what the reference is that we're trying to use, and then we're going to give it the reads. So the reads are in this FASTQ format normally. And so these are the reads that you would get back from the sequencing facility generally. And we're creating this output file, which is a SAM file. And so in the context of our course, or in your prior experience, you may have used other programs to map reads. So in the course, this step, we used HiSat2, for example. There are other tools that can do this, but in this case, we're using bwa mem. That's what's taking, that's what's fulfilling that role here. The other steps that are going on in this script are similar to what Susie described earlier, or was discussing earlier, which is converting SAM into ultimately a sorted BAM file, right? Because the SAM files take up a lot of space. They're nice because they're human readable, but in terms of conserving space and sort of running downstream steps, we want to basically convert those from these SAM files into BAM files. And so the script will run for probably a couple of hours when you try that, and it'll basically go through I think it's 14 different samples that are present, and do the read mapping for each one, and then do the file conversions, right? The next step is where we call the SNPs. And in this particular example, we're using Stacks to do that. So it's this program called Stacks, which is in the opt directory on your Atmosphere instance. And so in the context of our BCBC course, this step, the SNP calling proper, we used something called GATK. Again, there are a lot of programs that can do the SNP calling. Stacks is what's kind of fulfilling that that part here. This is the main kind of pipeline command. So Stacks has this script called refmap.pl. So the .pl indicates it's a Perl script, or should be a Perl script. And there are a number of steps in terms of what's done here. I'll point you to the documentation of Stacks for all the steps that it does. But essentially it's going to yield from our mapped reads, it's going to give us information on the variants. So at a given site in the genome, are individuals sort of the same as the reference genome at that site? Is there some variant there? And if there is a variant there, is it present in a homozygous state or heterozygous state? And so here we have to give it a path to our sample files, which are the end result of that previous mapping script step. So those will be sorted BAM files at this point, placed into a directory called sorted BAM. Stacks also asks for this population map.txt file, which is essentially a 2-column, usually I think it's a 2-column file with sort of the sample IDs and a priori population designation, right? And that can be altered, and it can be run in different ways for a real sample if you'd like. Again, the threads and our output directory, we're calling this ref snips, which is where we want to point our results. In what follows, so one thing I should mention here, so this ref_map.pl, if you were working with without a reference genome, I think there's a separate script called de novo PL, or I forget the exact name, but there's basically a parallel script that you can run if you don't have a reference, but you still want to sort of cluster the reads that you have and attempt to call SNPs from those clusters. And so this second command here is essentially using this populations program within stacks. In this case, it's just being used to export a VCF file. And a VCF file is essentially just a variant file that has all the SNPs, the various locations of SNPs in each of the samples. The other thing to point out here is that one of the things that Stacks is nice for, and I mean I've only started to use Stacks really. It does have the ability to output lots of different formats. So in what follows, we're working with a VCF file, which is a common format, and for the purposes of what follows in terms of the filtering, it's nice to use for this exercise. But for a number of kind of downstream programs or options, Stack seems to have this nice ability to, or it's been tailored to provide output that's already in a form that downstream programs are expecting. So for example, structure is a program that's fairly commonly used. There's also an R package called Adigenet, or that's how I pronounce it. Those programs all sort of expect slightly different inputs, and Stacks is pretty flexible in terms of providing different types of outputs. So I just wanted to highlight that here. Here we're using it to export a VCF, but there's Stacks seems pretty nice in terms of its flexibility, in terms of what it can kind of pre-process for you. The third kind of component, so the first step is mapping the reads to the reference, and the second is calling SNPs, right? In the context of our course, we did those things as well, right? We used slightly different programs, but we already kind of covered those in the intro course. Here, this is actually an extension that goes a bit beyond what we discussed in our SNP calling section of the course, where here we're going to filter SNPs using this VCF tools program. So there's a couple of commands here for installing VCF tools locally on your Atmosphere instance. Won't spend any time on those really. They should run fairly smoothly. They did for me yesterday. But if not, please feel free to message us or message me. Once you've installed it, the next commands are basically just working through different examples of how to filter your SNPs. So the VCF file that's initially generated is sometimes what we would call our raw VCF or our raw SNP file. That's what is generated from the SNP calling. And most SNP callers do, a fairly good job in terms of calling SNPs. But that is kind of if you take that as your starting set, it's sort of your most permissive or your broadest set of the possible SNPs in your data set. So you can do this sort of additional filtering using a tool like VCF tools. And so from that previous, the previous SNP calling steps, we ended up with this VCF file called populations.snips.vcf. Okay, and so that's the input in these two cases that's being used, and we're working with that VCF, and we're generating a more stringently filtered version of that. In one case, it's called UGIBA SNPs filtered. In this one, it's called UGIBA first pass down here. So this out is what determines what the prefix of the file, the output file will be. And essentially, VCF tools is a very flexible tool for filtering. So there's a lot of different parameters you can use to filter. Here are some kind of maybe more commonly used ones, things that are good to keep in mind. So in this example, it's, I guess the first thing I wanted to clarify here is we're filtering first based on sites, right? So we're filtering by site. We're looking at each SNP and we're sort of going along and calculating percentages across individuals, but we're filtering out by site. So we're filtering out certain sites. This first parameter is basically ensuring that we only keep SNPs, or basically it's going to throw out all SNPs that are missing more than 40% in 40% of the samples, is the way to think of that. It's going to filter out sites that have a depth below 3 or above 100. It's going to only keep things that have a minor allele frequency of 0.05% or above. And this one, the MAC, is the minor allele count, I think, across all individuals. The missing data parameter is used here again. So this is, I think, 60% or above is going to be thrown out. This one's always tricky because I think it's the inverse of what the actual number is. And here, min alleles and max alleles is being used to essentially extract only biallelic SNPs. So SNPs for which there are 2 alleles. So these are kind of examples of different parameters that you would want to adjust or tweak to give you sort of different levels of stringency in terms of the sites that you keep, the SNPs. And you can actually run these all together in one command. But the other thing I wanted to mention here is you can actually run them sequentially. So sometimes you'll sort of run one, maybe the max missing parameter, and then using that output, you can run a second layer on them. So you can sort of run them step by step and sort of see what the net effect is of each parameter on your number of SNPs remaining. These other commands, so VCF tools can also do a number of things in terms of getting you stats on different, on the samples that you have. So for example, if you use this --het option and run it on your VCF, here we're using one of the filtered VCFs, it'll give you the observed and expected heterozygosity for the samples in that file. It can also, in this case, the --− depth is giving you the average depth across the samples. This one I'll focus on a little bit because we're going to use it in the next step. And if you use this ---- missing INDV, it's actually going to calculate the proportion of SNPs, loci, missing by individual. Right? So before, we were filtering by site. So we were keeping certain sites that met certain criteria that we had. In the next step, we're actually going to filter out individuals that have too much missing data, right? So if there's an individual that doesn't have data for 90% of the SNPs in our VCF, we'll probably exclude that one because you know, it's possible something didn't go well with the sequencing. And so the way to do that, so this is a little bit more involved, and you might have to go through it a couple of times, is we're first going to use this to generate this file. So it's going to give us a file back, a results file called out.imis is the default. And that file is going to contain information about the, actually, let's see if I can pull up. I won't have the atmosphere version. Just try and pull something up quickly for illustrative purposes. So, so I have this file called out.imis, and that's what will be produced by the missing INDV by this command, the previous slide here, by using this parameter in VCF tools. And so if you don't add anything else to the name of the file, it'll just be called out.imis. And so if we look inside this type of file, you'll see it'll have the first column populated with whatever identifier you have. And the main column that we want to look at is this f_miss. And this is the frequency or the fraction of data that is missing from the file. So if you have, so per sample for a given sample, it's the proportion of the SNPs that are not present or not called in that file. And so using that information, we're gonna, so this is a little bit tricky here, but we're using this command called awk. And awk is going to select based on the fifth column. So the $5 relates to the fifth column in that file, which was our f_miss. And here we're going to select anything that has more than a fraction of 0.5, right? So if it's missing more than half of the data. So if we go here, you can see that this is column 5, 1, 2, 3, 4, 5. And so anything that is greater than 0.5, that line from that file is going to get extracted by this part of the command. I can actually, let's try and demonstrate that here. So I'm going to do awk $5 greater than 0.5. So you can see the result of that part of the command is to extract all the samples, all the lines relating to samples that have more than 50% missing data in this case, right? And we could have adjusted this, right? We could have said 80%, right? Just by changing that command. Now here what we're doing is a pipe, which hopefully some of you have seen before or remember. And we're basically taking that output and we're passing it into a cut command. And the cut command is going to extract the first column of that file, or sorry, the first column of that output from this command. Okay, so again, we're piping information from this first command, this awk command that I've done. And we're going to transfer it into cut minus F1. And so the result of that is it's going to give me basically all the sample IDs for any sample that is missing too much data, right, is above a given threshold. And we're going to save it into this file here called lowdp50.imdb. The upshot of that is that we can then take that list of samples and use that to refilter using VCF tools using this parameter called remove. And with remove, you pass it a set of sample IDs of things that you want to just exclude from your VCF, right? And so if we pass it that list, it's going to remove all those individuals from our final VCF, okay? So effectively, we've kind of combined a couple of commands there to filter not by site this time, but also by individual, by sample on a per sample basis. So again, filtering filtering individuals that have too much missing data. And that's in addition to the filtering that we did where certain sites had too much missing data on a per sample basis. The last little bit to highlight here is there's an R script included in this, this directory that you can use to run and generate a PCA. So run a PCA analysis and generate a plot. I would recommend a couple of things with this. So as before, the script itself is just a text file, so you can open it up and take a look and see sort of the particular steps that it's doing. And of course, the neighbor is mowing the lawn. Hopefully you can't hear that too much. Amazing. Okay, so one, you can just sort of run the script as is. However, I would recommend actually downloading the data file and the script to your own computer and maybe opening it in RStudio, which some of our students have seen before and others might be familiar with. And you can check it out on our class materials. Because RStudio is a nice kind of graphical user interface where you can explore the script and see what's going on inside it. The other thing to note is that this script is built to work with the data set that was included in Atmosphere. So it's specifically tailored to the VCF file that you'll have generated from these other steps when you run through the module. If you wanted to use the script on your own data, you're going to want to open that script and change the lines where that VCF file is kind of hard-coded, as we'd say. So if you have a different VCF file with your own data, you would want to maybe go in there and adjust that. And you can do that in either RStudio or in a text editor of your choice. And I guess the last thing that I did want to highlight with relation to that script is if you look inside it, there's an R function, and we're not, I guess, going to go into a lot about R, but there's a function being used in there that is actually doing some LD pruning before it runs the principal component analysis. So it's pruning the SNPs that have been loaded into R based on their linkage to superequilibrium. So assessing two SNPs, and if there is too high a correlation between them, it sort of excludes one of that pair, right? And I guess that's just an example I want to highlight that even though the module includes these VCF tool filtering steps, and that's like a really nice way of doing things and keeping things clean, a lot of the times you can also incorporate, or a lot of the PopGen tools in R, for example, will include additional functions for filtering, additional filtering that can be done. So it's sort of this sort of highlights the different options that you have in terms of the types of filtering that you can do. Ultimately, it depends on kind of the pipeline and how you feel it's best to streamline things in terms of your workflow. Certainly, VCF tools is handy and you can use for a lot of the filtering steps. But the R packages themselves that you would be doing a lot of PopGen analysis within or using for the PopGen analyses in R have a lot of these kind of additional filtering steps or functions available within them. And yeah, so with that, I'll basically end there. Maybe we end a bit early. But yeah, so the main kind of components again of this module. First step, again, mapping reads to your reference genome. In this case, we're using BWA-MEM to achieve that. The second step is the SNP calling. In this module, it's stacks that's being used, taking the place of GATK or other SNP callers. Then there are a couple of filtering steps. And so you can use VCF tools to explore kind of different parameters, different options for kind of removing, particularly removing loci and individuals that have too much missing data. And then you can load your VCF file into R for generating PCA plot and other types of PopGen analyses. With again the reminder that Stacks also has the capability, if you're using a different R package that has slightly different, I mean, a lot of them have different sort of file format expectations. That Stacks has this nice ability in most cases to generate the types of files that these other R packages might be expecting. And I will end there. Yeah, I guess the only other thing to mention is if you are running through these on your Atmosphere instance, you might need to have it open and running for a little while. I don't know. I guess you could sort of use Screen or NoHup maybe to disconnect from Atmosphere. We don't really go into it too much. But otherwise, I mean, it should work even if you just sort of have it open for a little while. So yeah, feel free to sort of take a look through that and work through it if you have a bit of time, and let myself or Jacob know if any questions come up. But like I said, check the PDF. The module that Jacob developed is really nice. It has a lot of sort of the detailed instructions and discussion of the different steps. And the more times you see these things, the more they become internalized. Give them time to decant a bit.
The one thing I would add, Adrian, on the filtering that you mentioned about doing the piecemeal, like to do like one step at a time. I've noticed that if you do it that way, sometimes you get different results than if you do them all at the same time. A lot like Mazurka with the assemblies, where you could run the same code time and time again, and you may not get the exact same answer every time. You'd be close, but not necessarily get 100% the same answer every time. But just to keep that in mind if you're filtering things.
Yeah, and I think, I mean, yeah, that's a really good point. You also have to be careful the order in which you do things if you do them sequentially, because sometimes certain functions don't actually throw things out. They rather mark things as missing. So for example, if something is below a certain depth, it's not that that site will be removed, but for any sample that has that site below the given depth, it'll just mark it as a dot/dot rather than, it won't actually remove that site because it can't remove an entire row because it might be present for other samples. So if you do things in that order, it'll affect the number of the amount of missing data for that individual, right? So depending on the order that you do things in, it'll definitely change what you get. So maybe some caution is warranted as well. But I mean, what's nice about VCF tool is like if you're running it on a server, it doesn't take too long to run usually. And you know, you can try different sort of different runs and different versions and sort of see how it's behaving. Sometimes it's nice to sort of plot how many SNPs you have after each sort of filtering step and see how it's behaving, see how it's treating your VCF file, your SNPs. Yeah, no, that's a really good point. I guess any other comments?
Can I also mention that you can import VCF files into Koji and view them in the context of your favorite genome?
Yeah, fair. And I think, I mean, again, I keep referencing our course, which isn't fair to all those who didn't attend it, but you can stay tuned for future versions of the course. But I think as we've discussed also, VCF, even though a lot of these R packages do require or often request their own or particular types of formats, VCF is kind of if you're not familiar with it, it is kind of one of the standard forms for representing SNP data now. Things have sort of converged quite a bit. So yeah, generating the VCF file will get you a long way, and we'll let you do even more in Koji. And I guess, I don't know if, does Susie have any concluding
I was just
remarks or something?
If there were any more
If there are any questions, yeah.
Thank you, Adrian. Are there any more questions for Adrian? All right, well, I think that's it for our workshop. I want to thank all of the speakers so much for participating, and all of the participants. You've been a great group. I hope that you're able to take some of these things that we've provided today and move forward with your own research. And you can always feel free to reach out to any of us if you need, you know, any help with anything, questions. I'll put our course that Adrian mentioned in the chat, and where's the chat? We typically do this in the spring. So if anyone wants to join our mailing list, if you're not at BTI, you can send us an email. So my email is srs57@cornell.edu. I'm putting that in the chat too. So, you know, if you want, we can certainly tell you about upcoming things that we might be doing that, you know, maybe you'd be interested in. I want to thank everyone at Cyverse again for helping us get these VMs up and running, and, you know, all of the work that we're doing is just so much easier to be able to run that on their infrastructure, as opposed to hacking together something on our local servers, which would be an utter disaster. So they are greatly appreciated. Anything else? I think that's about it. And like I said, feel free to get in touch. We look forward to chatting with you more. I hope, yeah.
Sorry, one other kind of administrative thing. I think when you aren't using your Atmosphere VM, it's a good idea to suspend it. I don't think we mentioned that at all before, but actually quite important. If you go to the site where it has your instance and where you sort of open the web shell, there's a button there that says suspend, and I would encourage you to suspend it if you aren't going to be working with it for a while.
Otherwise, it'll use up all your resources, and then you'll have to request more. So if you plan on using it again, definitely. I mean, regardless, you should suspend it. But yeah, I think that's it. I hope you enjoy the rest of your day wherever you are, and take care.